I would like to welcome everyone once again. So myself, Nityan and Sharma, and I'm a senior product manager in Premas Life Sciences, and I take care of this 10x Genomics portfolio, which is a company which is based in US, and they are the global leader in this area. So using their workflow and other stuff, we will try to learn what exactly this application is, and then we can take up some questions from your end to answer. I will be happy to answer them. So let's start the presentation. Let me use my laser pointer. Okay. So all tissues are heterogeneous and complex. So that is the foremost, we can say, complexity that is we observe when we see any tissue sample which is coming from human, and which is common to all the organisms which are multicellular in nature. And if you see, every cell type in a tissue, in an organ, has a specific role. And when there is a deviation in those roles, then it leads to some specific diseases. Or if there is some deficiency of particular cell type function, then also we encounter various diseases. So whatever data right now we are generating is the genomic data is almost constant, what we say. So that is like Mendelian inheritance or diseases causing by a particular point mutation can be picked up by NGS technologies, and we can say that yes. But the real aspect is the functional aspects. So functional aspect is happening at RNA and protein level, where the enzymes are being synthesized or functional units are there that are going to do the metabolic activities, and those deviation leads to the diseases. So if you can go through the Human Cell Atlas program, this is a program initiated by Science Zuckerberg Initiative that is to catalog all the human cells present in a body, and also find out the causes of different diseases that are encountered in the human, we can say, population. So this is where the single cell technologies are helping to these initiatives so that the right type of data can be generated. Now, I think from Udit's presentation and from Ashutosh's presentation, you people understood that we have to sequence a DNA element, or we can say DNA entity. So here you can see the forward read and the reverse read. That means something we are sequencing from this end, from this end. And to sequence that, what we need. So on Illumina sequencer, we need two adapters on both the ends. We can see here read one primer, read two primer. So that will typically leads to the sequencing of the insert or the DNA insert. Then we have I5 and I7 indexes. So these indexes are useful when you want to do multiple samples in a one go. So this index reads will help you to demultiplex the data. And apart from this, you can also see the P5 and P7 primer adapters. So these adapters are inherent part of the Illumina sequencer, where they will help those DNA elements binding to the flow cell and performing the clonal amplification, which Udit has just shown in the video. Now, right now, we are able to segregate data sample-wise, means whatever read coming from which sample. Now, if you want to sequence it to the cell, then you can see there is, I have to add a cell barcode to that element. So this is like the need. Then I will add the other adapters upstream and the downstream of those sequences. So now I'll come to know which sample the read is coming and from which cell the read is coming. So sample also demultiplexing, and the cell data is also getting demultiplexed. So this is the purpose of the single cell technology. And at the same time, your need is to count the RNA that we have captured. So the counting is happening based on unique molecular identifier, and rest of the element will be same. So this is going to help you to tag whatever RNA you capture from the sample, give you a unique molecular tag so that the PCR artifact could be removed, and easily we can do the counting part. So ultimately what you will have, how many molecule we captured, from which cell you captured, and from which sample the data is coming. And these are the sequencing read that is the standard to the Illumina sequencing chemistry. So which gene, how many copy of those gene or molecule we captured, from which cell we captured, and from which sample we are capturing. So sample indexes are just to multiplex multiple samples for sequencing run. Hope this is almost clear to everyone. Now, how the data looks like on a Disney plot. This is a plot where you can see different dots are there. So each dot is a cell, and based on their gene expression pattern or closer, they will be clustered on this Disney plot. And ultimately, you can see erythrocytes are there, some neurons are there, and all the neurons have different clusters. So that means they do different functions and they have differences in their gene expression pattern. So this data is coming from 10,000 mouse brain derived from the cortex, hippocampus, ventricular zones, and embryon from the embryonic day 18, and all the major subpopulation we are able to pick it up. Now, if it is a disease sample, you will be able to see some deviations in the gene expression pattern. And the disease-specific gene will be expressing in some of the population, not all the population. So this is like we are adding one more dimension to my NGS data, that is from which cell the RNA is coming. Now, it is like you have to do tissue dissociation to get the single cell suspension. Now, people working on tissues demanded that no, we don't want to disrupt the tissue. We want to map the data on the tissue image only, or the tissue surface, or the tissue section only. So then another technology that is again from tennis genomics is helping to map the data to the location on the tissue at 1 to 10 cell resolution right now, which is like in the next year, you will get a single cell resolution. So here, if you see, this is a histology image. Many people are aware of sectioning the tissue, placing on the slide, doing imaging or H&E staining, and then taking the image, and then morphologically find out what cell types, which tissue type, where exactly the variation is. And pathologists see where the cancer cell is, or oncopathologists see where the cancer and normal cells, how the interactions are happening. But that is just based on those morphological features. But ultimately, what is happening at the gene expression level, we are limited to one or two or even three or four markers only. Not we are not getting the story of rest of the RNA, what they are doing, or rest of the gene, what they are doing at different location. So now this technology is going to help you capture the RNA spot, means location wise, and giving a specific barcode like the cell barcode we are giving. And ultimately, when you sequence the data, you will be able to map it to the location. So here you can see the different like cell types, or we can say tissue types, are clustering together, and ultimately all these clusters are going to be mapped at different location. So you can see the feature or the expression feature of this image is matching the morphological image. That means the technology is robust and is able to deliver you the right type of data and able to capture the right amount of RNA from every spot, and then map it to the location based on the using the bioinformatics software. Now, it has been already understood by the scientific community that these technology will bring a big promise in the clinical domain, I can say, and that's why science has recognized this method as a breakthrough of the year in 2018. And when multiple data from the same sample you are generating, like some chromatin data, some genome data, protein data, methylation data, transcriptome data from the same cell, that was recognized as the nature's method of the year in 2019. And already this technology is being I think the scientists has recognized is at the top 10 innovation from 2015 to 20 onwards. Every year there is a new addition to this portfolio, and it became the method of the rated as the top innovation by the Scientist magazine. So you'll be seeing some more products from Tennis Genomics, which will be rated at top 10 innovations every year. The publication number is already increasing exponentially, I can say, and right now 3,000 plus publications are there, mostly in top-rated journals, Nature, Cell, Science. So the people are generating the right type of data from the right type of sample, and right biological story they are able to decipher from their samples, and ultimately they are getting published in high-rated journals. Now, when it comes to the story of where how far is the clinic story and how far it is NGS technologies or the single cell technologies will be hitting the clinics. So ultimately, the story always start with the R&D or research, and definitely many labs have adopted these technologies, including the pharma companies, and we are able to see that some inputs and outputs are getting generated from these new technological from this new technological innovations, and you can able to see that better profiling or of the cancer samples we are able to do. We are able to understand what exactly transcriptome differences are happening in the relapse. Even if we are doing a cell-based therapy, what is the performance of those cell which are infused in the patient? So those things we can monitor using these technologies. So ultimately, yes, the story has already started, and some publications are in this area, and people are trying to generate or standardize this technology so that they can offer a specific test. And some companies are there in this area at the same time. Now, coming to the drug discovery journey, which is the clinical aspect, what we know. Starting from its first objective target identification, the technology is going to help in a bigger way, because we are able to catalog each and every cell and what exactly it is doing in that sample. So we are able to understand the targets. Then performance of the drugs in terms of preclinical studies. So we are able to do those aspects. So you can see some publications and understanding the T cell expansion in the tumor infiltration, the clinical response, and some here you can see the clinically relevant core needle biopsies for evaluation of tumor immune cell interaction. So all this is based on a single cell sequencing technologies people are doing. Ultimately, clinical research is there, and the clinical trial is also there at the same time. So preclinical, clinical research and trials. In every phase, you can see this technology is going to help the user to generate the right type of data, what they need for their experiments. Now, as I told already, the broad adoption is there from all the top 20 biopharma companies, which right now you can see on the screen. You are able to, you can just pick up Bristol Myers Squibb, Novartis, GSK is the biggest brand. Roche is already a very big brand. Genentech. The Roche is like, even though they don't have these technologies, but they adopt the technology which are upcoming and have bigger promise in the pharma domain, and push their R&D in those directions. Similarly, AstraZeneca is there, Sanofi is there, Lilly is there, Merck is there, Takeda, Biogen, Boehringer, Ham is there, and the Novo Nordisk. So all those big pharma companies have already invested in these methods to generate the clinical data from their trials. Now, at the same time, this single cell technology is going to replace and complement, I will say, to the traditional methods by which we wanted to understand the genomics. We want to do the CRISPR screening. We want to do RNA sequencing. We want to do T cell, B cell receptor studies. We want to perform chromatin studies, and we want to do protein expression studies. So there are different various methods that is being used. Now, one workflow is a single cell technology, which can help you generate this diverse data using an NGS sequencer in a more simpler way, as compared to multiple platforms, multiple equipment you are using to generate different type of data from the sample. And it has also been promising, or has promised that you can generate 2 or 3 types of data, or 4 types of data from the same cell which practically impossible by any other technology right now. Now, coming to the complete solution, what we have in the single cell area is the instrument. So you need a specific instrument so that the single cell library can be prepared. And this equipment needs the consumable part, which is also provided and manufactured by 10S Genomics. You can see different reagents are there. The chip is there, and this is for Visium slides to generate the data from the tissue section. And then we have the software part. So once the data is out, how to analyze and visualize the data. So this part is totally free from 10S Genomics. Anyone can download the software and install it and learn the pick some data sets and start analyzing the data at the same time. So there is no need to get any license for these softwares. Now, as I told that we are able to do multiple aspects of a biological sample, in which you can see there are publication in the area of CRISPR. You can see multiple publication. You can see area epigenomics, where you can see some publications there. The gene expression is already the story started with this, and you can see multiple applications or publications are there where people are generating data from the RNA of individual cells. And then you can see high throughput immune profiling, which is also right now in the COVID area very, very important. And using these technology, they got some breakthrough. And apart from this, the high throughput protein screening, you can see some publications are there where some erythrosclerotic plaques and some other systemic lupus is there. So people are generating what the type of data which was needed previously because of technical limitations, they were not generating, but now the technological limitations has been overcome and people are exploring all those aspects of the biological science. So here you can see acquired cancer resistance was understood by the transcriptional loss of class 1 HLA. So all those aspects are very, very important, and single cell technology is going to help in that area. Now, how exactly we have to generate this huge biological data at single cell resolution? So what is the background behind this? So if you see 10x product lines are basically fall into 4 categories in the single cell. One is gene expression, immune profiling, attack, and attack plus gene expression. So this gene expression and immune profiling have more additional feature in which you are able to do cell surface protein. And also we offer a targeted panel so that you can save the cost on sequencing part, because sequencing the whole transcriptome sometimes is a challenging when you want to limit your budget. So in that way, the targeted panel can reduce the sequencing cost up to 80%. Now, cell surface protein means you can generate the data of the protein marker or cell surface marker similar to what people are generating using flow data. So flow and genomic data or transcriptome data from the same sample can be generated in a one workflow. Then people are in the area of CRISPR profiling, having perturbations to be done because of those CRISPR guide RNA, what gene expression changes are happening. So we are able to link both the data in a single shot. So CRISPR screening is more streamlined and more productive, I can say. And then we have CellPlex for gene expression product line. So in this, the CellPlex assay, you can pool up to 12 samples in one reaction. So you will be able to save the cost of library preparation when you are doing a CellPlex assay. At the same time, limiting the number of cells, but some applications don't need screening of lot many cells. So such applications can be easily fitted, and also you can save the cost. Then coming the antigen specificity, which is a very much important aspect of any T cell. So my TCR sequence or this T cell is binding to which antigen. So this is feasible by another company, Immudex, that provides the reagents of a class 1, class 2 represented antigens, MST represented antigen, the Dextramer technology. So once you incubate this with the T cell population, so we come to know which TCR are binding to our specific antigens. Now, this is happening. The data is being captured by the beads. So these beads are very, very important. The gel beads, what we, the 10S technology. So how these primers, so this is like a primer delivery agent you can call. And these beads have specific primers for every application. So if you see the gene expression one, so in the end, you can see there are three primers are there, but in the end, you can see the poly DT, capture sequence one and capture sequence two. So poly DT is definitely is going to capture the mRNA. Capture sequence one and two, sorry, are going to capture the CRISPR data or the cell surface data of the same cell. And the UMI, I already explained, that is going to do the counting application. And 10S barcode is going to help you to demultiplex the data at individual cell level. So from which cell the that data is captured, or that molecule is captured, that we come to know. And read one will be the part of the Illumina primers. So likewise, these primers are delivered to individual cells, and ultimately they are going to capture the molecules, RNA or those CRISPR or those cell surface protein DNA barcodes. Now, this is happening at the 3' end. In the immune profiling, since we are sequencing TCR and the BCR, our purpose is to attach the 10x barcode and the UMI to the 5' end of the mRNA, which is feasible by the target switch oligo technology. So this is going to help to attach at the 5' end, so that we can sequence the full length TCR and the BCR of the immune cells, that is B cells and the T cells. Now for the attack data, that is the genomic data, that is also a dynamic. We can say when a cell is working at the genome level, some portions of those DNA are open and some are closed, euchromatin and heterochromatin, what we can call in a simple term. So whatever open regions are there, so those signals are being captured by this read one complementary DNA sequences, and we are using a transposase technology to cut down and tag those open chromatin region of individual nuclei. And ultimately, you will be able to generate at single cell resolution the epigenomics of that particular cell. And last one is attack plus gene expression, which was launched last year, and many publications are coming up within this, because it is able to capture the chromatin of the same nuclei and the RNA of the same nuclei. So the epigenomic landscape of the same cell and transcriptomic landscape of the same cell. So how they are interrelated, and how correlations can be made very easily without any difficult bioinformatics pipeline. So such new solution is welcomed by the scientific community, and many data has been generated using these technology. So thing to remember here is we are capturing the molecules from a cell using a bead. So this is a functional bead, and every application has a specific molecular signatures on their beads. Now, where exactly you have to use these reagents? So this reagent to be used on either Chromium controller or Chromium X or IX, where I can see that it is going to do low throughput and standard throughput, and we'll be coming to know what exactly low throughput, standard throughput, and high throughput technology is. So Chromium X going to support the HT kits, and IX need to be upgraded so that it can do HT kits. Now, this slide will give you an idea about what exactly we have to do to convert cells into libraries or sequencing libraries. So first job is to prepare the cell suspension. So we have to do counting, we have to do QC of the cell, whether they are what we say exactly in terms of viability of those cells, the number of cells, the size of the cells, the debris. So all those things, we say that it should be debris free. The appropriate amount of cells has to be there for every sample. And then we prepare enzyme mix similar to RT enzyme mix, and we add the counted cell into that particular sample. In the meantime, we have to assemble the chip on a aluminum cassette. So here you can see that there are 8 channels. So this means we can process 8 samples at a time. And you can see there are row 1, 2, and 3. So this indicates what exactly, where exactly we have to load the reagents. So first job is to prepare the cell and enzyme mix and load it in the row 1. So all the 8 samples have been loaded. Now in the row 2, you can add the gel beads. So these are individually packed gel beads. So what we see in the previous slide, the gel beads, and ultimately you will be able to add in the row 2, or you have to add, and in the top well, you will be loading the oil. So this is like the whole pipetting step that you have to do on this particular chip. And if you don't want to use all the wells, so you can fill the rest of the wells or empty wells with the 50% glycerol. And then you'll have to add a gasket on this. This is again provided by 10x. And ultimately, you'll be placing it in either Chromium controller or in a Chromium X system or IX system. And you can see here what exactly is happening in a video. The beads are moving, the reagents are going to be mixed, and ultimately one cell is going to captured randomly in these reactions. So this is the cell that you just saw that got captured in that reaction. So this is happening continuously, and millions of partitions are being created, and few of them will have cells. So like if you have loaded 16,000 cells or 17,000 cells, then almost 10,000 partitions or these reactions will have cells, and rest of will be empty, so that we can remove the doublet efficiency of this particular workflow. Because if you load more cells, the chances of 2 cells in one reaction will be high. So to reduce that, we generate sufficiently large amount of partitions or empty partitions, and ultimately you are able to see that cell is getting captured randomly in these reactions. And everyone has its own 10x barcode, so we are able to tag the molecules of those cells. Now, this slide is ultimately the biochemistry of the workflow, you can say. So likewise, you will be able to see, if you see under microscope, the cells are captured, the bead is there, and you can see here the cell is also there. So this is the reaction entity. It has RT enzyme, other lysis reagents, which are going to lyse these beads in the cells, and ultimately all the primers and the RNA will be in a reaction, or we can say independent reaction. And then we have to collect it and take it in a PCR tube and do a RT-PCR step. So in this step only, the content of the reactions are getting barcoded. So once you barcode, then you can use a recovery agent. You will be using a recovery agent so that it can be disrupted, and the pool of the barcoded cDNA will be purified, and ultimately you will be converting into a sequenceable library, which we have just discussed. These are the features of those sequencer libraries. So you can see 10s barcode, EMI, the poly DT, how what we captured, and from the other end we are reading the sequence of the gene. So we come to know which gene it is, which sample from the indexes, how many copy of those molecule we captured, and 10s barcode tell us from which cell it is captured. So ultimately that becomes the input on the Illumina machine. Here you can see NextSeq, NovaSeq, but it is compatible with all the platforms. But depending on the amount of data, you can choose the right platform. Now, once the data is out, then you will have to use a cell ranger pipeline, where you can see ultimate goal is to catalog the cell numbers, the gene, and the feature if you capture some feature, and ultimately do a counting in the cell and put across like how many molecule we captured for each cell, for each gene. And ultimately this will be very huge matrix. And to simplify it in a visualization way, you will using the loop browser. The output file is a cloop file, which will be used on this software, so that you can see the what type of cells are present in a sample. And these are some markers that let you know catalog those cells or name those cells. And you can see the T cell tumor cells also. We can see some marker top 2A, BRIC5 and CCNA2. NK cells are there, monocytes are there, exhausted helper T cells are there, and the normal T cells are there, which has CD3E marker. So ultimately, you will be able to see the whole heterogeneity of your sample type depending on the sample you are working on. Now, the next slide is the summary of the single cell workflow. So starting material will be a cell on the nuclei or nuclei. You have the system to do the library preparation. So you have various kits. Depending on your experiment, you have to choose either any one of the kits from this. Then once you prepare the library, you will be doing the sequencing part. And then comes the data analysis, where Cell Ranger and Loop Browser are going to help you the data analysis. Now let's see some experiments. Before that, I think Chromium X update I want to give you. So the Chromium controller or standard system, it will be capturing 10,000 cells maximum per channel. Whereas the Chromium X with their ST kit will be able to help you capture up to 20,000 cells per channel. And at the same time, 16 samples at a time instead of 8 samples at a time, which I just saw in the previous slide. And the capture efficiency is still the 65%. Multiplet rate is low, around 0.5% for 1,000 cells. And it is able to reduce the overall cost of the project by 60%. And if you're doing a cell multiplexing, you are able to do up to 750,000 cells in a one run. So it is very much highly like flexible system which supports the existing kits also and the new ST kits. So how it is achieved? So they have do a smart work where the gel beads are being used for the 2A and 2B. These two are the sample rows, and gel bead is common. And the top well and the bottom well are the collection wells. And ultimately, you are able to see that in this diagrammatic way, the gel bead 1 is going to 4 channels, and enzymes or cell mix is moving through 2 channels, and ultimately these 2 channels are ending up in the top and the bottom well. So 10,000 cells from one end, and from the other end also 10,000 cells. So total 20,000 cells we are capturing in both the top and the bottom recovery wells. So this way they are achieving doubling the number of samples per channel and also doubling the number of cells captured per channel. So ultimately, it is 4 times we are able to do the capturing number of cells. Now, as I told that LT low throughput kit is there, standard kit is there, ST kit is there, Celplex kit is there, and Celplex HT kit is there. So if you see, the low throughput is supporting 1,000 cells per channel, standard is supporting 10,000 maximum, ST is supporting 20,000. And if you're doing Celplex on a standard workflow, you're able to do 30,000 cells per channel. And same thing, if you're doing Celplex on HT chip, you are still doubling up to 60,000 cells per channel. Now, in terms of total capacity of the chip, like LT can do 8 samples, that means 8,000 cells per run. Standard can do again 8,000, then it is 80,000 cells. And if you're doing HT1, so it can do 3,20,000 cells. And if you're doing Celplex standard, that will lead to 240,000 cells. And if you're doing Celplex HT, that will give you 960,000 cells. That means close to 1,000,000 cells. So that is like 1,000,000 cell story you can create using like in a very short span of time using Chromium X system with Celplex assay. Now, as the things are moving forward, what is we have observed or 10x has observed as before 2005, people were publishing data of 100 cells and more than that, and then slowly 2010, 15, the story had just started where people are doing many cell samples. We can say million cell samples are coming up in this after 2020, between 2019 and 2020, and it's still going on. And at the same time, the number of reported cell types are also going increasing. When you catalog more number of cells, definitely you will find more rare cell population. So those findings are also coming up. So ultimately, the demand or the user needs these methods so that they can catalog each and every cell of their sample in a cost effective way. So this 10x is going to help in that way. Now, where is the real application if you want to see the discovery, translational, and a clinical research? If you see, this is data of a cancer sample, lung cancer sample, where you can see there is a 1, 2, 3, and 4. These are 4 different types of sample there. So you can see the yellow one on the category, the different color. So that means they have some differences in the cell types. And ultimately, if you see the cell type based on their whatever function is there, so you can say cancer one. So here is also cancer, here also cancer, and these are the myeloid population. So if you see these two cancers are very different and coming from two different samples. So that means the heterogeneity is very huge. What we wanted to understand of any cancer sample. Then in the developmental biology stage or any developmental functional aspects, you can see from where the cells are going and ending up to which cell type. So all those trajectories you can do and construct the developmental hierarchy and trace the disease evolution. At the same time, the cell lineage relationship can be done. And at the same time, functional aspects are there where in a particular specific cell population, you can see if you on and off a particular gene and do the treatment, pre-treatment, and then recurrence is there, then exactly why it is happening. So all those stories you can just understand using single cell technology. You'll be able to define the pathway, the network effects. You were able to do whatever discovery in terms of regulatory network are happening in a cell, and also correlate the specific phenotypes outcome, why this phenotype is happening. So ultimately, this is like going into more deeper and generating a right type of data from your samples. Now, this is an example where the need of the technology, why we need this technology is explanatory or self-explanatory, which was not evident by any other flow data or any other specific, because we don't have any marker for those rare cell populations. And that's why we need to generate data from whatever gene they're expressing. Simplest way to capture the RNA of every cell and then generate do the sequencing part. So here, this is the data of lung cell population, where what they were doing, they found a very rare cell population, which they termed as the inocytes. And when during the data analysis, they found this was only 5.5% of the cell. But the importance was there because they are contributing to the 50% of the lung CFTR. So whatever gene expression, suppose you're doing real-time PCR from this particular sample. So whatever curve is there, the 50% is contributed by this 0.5%. So the whole story of these people who were doing the CFTR clinical trials or drug development, who focused on the ciliated cells, now move to this cell population, and then now they will be focusing on the cell population to develop the drugs, because this is the expression center of those CFTR genes. So that is the story was turned upside down using this single cell technology, and ultimately that was established in mouse and human, where those cell population are in a very limited number, and the clinical trials and studies has been shifted completely to different direction as compared to what they were managing right now. So this is the importance of finding the rare cell population or the target cell population against which I want to develop a drug. Because now people are doing some research, or the research is mature based on antibody based delivery, but ultimately I want to see to which cell I want to deliver my drug, and what method I can use to deliver the drug, what kind of receptors are there on that particular cell. So this is going to be available using these technologies. Now in the COVID one, what this user have done is the ball fluid was used. So this ball fluid was ultimately collected from 9 to 18 days after first symptom, and three recovered mild cases were seen and six severe cases, and some publicly available single cell RNA sequencing data from eight normal lung samples were done. And ultimately, what exactly, if you see, this is the ball fluid from mild cohort and the severe cohort, and the 10x genomics was done. Single cell RNA data was generated. They also did the TCR, or the TCR sequencing of T cell population, and do the mathematical analysis. So this is the overall, if you see, they have grouped the 1, 2, and 3, 4. That means the different cell types. And ultimately, if you see, just by visualization effect, or just by visualizing the healthy control, the pattern of different cells are totally getting shift to some other cell types. And this here, the severe is totally different from the mild one. Though in the mild also there is a shift, but severe one, you can see there is a more recruitment of other cell types. So ultimately this means there something is happening at the cellular level, and then the different cell population are getting increased in the severe patient, and that is the cause of the severity of the disease. And when they also see the expression of specific genes that are responsible for what we say, so this one, what we call uh, uh, in terms of what technical term is sorry, I'm just avoiding this one, related to inflammations. So FC1 and
Nitya,
SPP1.
Nitya, take 2 minutes
Yeah.
break. You can have a water, then you speak. Have a water, then you speak. No problem.
Yes, this is a lot of information we are also getting, so it's all right. Thank you. Meanwhile, if anyone has any questions, please put it in the chat and we'll cover it towards the end of the session.
Yeah, I'm back.
Yes, go ahead. Yeah.
So these are the specific genes that they found that there was high expression in the severe patient. You can see the sixth patient, and this particular gene was not expressed in that particular patients, whereas they were completely expressed or highly expressed in the healthy and the mild one. So these are the markers that they found that are responsible for cytokine storm and other stuff that we found in the COVID severe patient. And when they analyzed the T cell populations, they found that the clonotype diversity, you can see the red one is only one clonotype, 2 to 5. You can see the green one, and more than 5 is the blue one. So in the moderate one, you can see there was expansion of those clones of CD, the T cell population, specifically expressing a specific markers, and that led to the we can say more equipped the patient is when the diversity is more to manage the infection. So ultimately, this was not happening in the severe cohort. We can see a very limited cell population that was there, but there was no clonotype expansion that was happening, which was required that particular to manage the disease. And moderate in the moderate patient, we are able to see that expansion happening. So a lot many stories and what exactly cell populations are working in a milder cohort and in the severe cohort. We can figure it out using the signal cell technology, which we were completely missing when we are doing normal NGS or normal flow or any other workflow where the single cell resolution of data was not getting generated. Now, coming to the next slide is a very important one, I can say. I will try to explain it to the listeners. It's, it's busy, but in a simple way. Just first you see this particular one where you can see antigen is there. There is a DNA barcode. There is a biotin and there is a streptavidin. So this is to purify those labeled cell population. So this is to be used to incubated with the B cell population that are expressing different antibodies, and then you can see that are getting added to the particular staining method or we have done, and then based on those PE fluoroprobe or phycoerythrin fluoroprobe, we are sorting those population, and ultimately this has to be taken as an input in the 10x workflow, and you are able to capture two data. One is what antigen is bound to the cell based on that barcode data we generate, and then we are able to sequence the antibody of that particular B cell. So we are doing the antibody sequencing and which antigen it is bound. So ultimately in this table, they have just shown a particular screening of the 9 antigens, HIV and flu, to find the novel neutralizing antibodies, which is now common for COVID also. If you see, they found that some antibody are binding to only one type of antigen. Some are binding to more than one antigens. So these are the number of those clonotypes they discovered. And ultimately, they identify 124 clones combining across 4 or more antigens. So these clones will be, we can say, pharmaceutically important because they have neutralizing effect on 4 and more antigens, and that can be used in a broader way to treat a specific or manage the specific disease because they have broad neutralizing effect. So only one antibody or clonotype can do the neutralization of HIV and flu at the same time, and if you can discover some other antigens also. So that is like a broad neutralizing antibody, what we call in technical term, on which people are working on such vaccine development program which can generate such broad neutralizing antibodies to in the human body, which can manage many pathogens as compared to, like, we have specificity also, but we need the broadness also. So this is the kind of research that people are doing, which is broadening up the immunological aspects of the vaccines, I can say. Now, as I told that we are able to multiplex the cells and then generate the data using the cellplex assay. So this is the data that is generated from 4 samples. So you can see adenocarcinoma, small cell lung carcinoma, squamous cell carcinoma, and lung cancer. A sample was there, and you can see multiplex and more tags that will be coming when we are doing loading more number of cells, but ultimately we can filter out them from our data. So first, if you see, just demultiplex the data based on the cell barcode, so you can easily demultiply the data into all the antibodies or cell surface protein markers. Around 17 markers were used. Then you are able to see the cell populations. So these are the various cell population. Here is the name of those cell population: B cell, dendritic, monocytes, monocyte CD16, NK, D double negative, TC effector, TC memory, TC naive, TH memory, TH naive, and undetermined. So this is like all the T cell population if you see. And non-small cell lung carcinoma is mostly B cell population is very high, which we are able to see here as compared to rest of the other cancer. And in this the undetermined lung cancer, you can see the T cell population is very high. That means this is not a non-small, this is something else, which is evident from this particular data set. And it is not also adenocarcinoma and small cell, squamous cell carcinoma. So we have more detailed view of our sample, I can say, when we are doing single cell technology. Now, as I told that we are able to capture two type of data from the same cell. One is the chromatin, the attack data, and other is the gene expression data. So here, this window, if you see, this window is technically representing this data or the gene expression data. And why it is important, I can say, from this group, which is a B cell population, which is naive and memory B cell, where they have the same expression pattern clustering together in this window, whereas they are getting splitted into the attack data into two sample types based on the memory and the naive, because the chromatin patterns is different. So ultimately, it is giving more power to resolve the data at different resolution. And this pattern, if you see, this pattern is coming for the maturation of the T cell population from the naive T cell population to the NK cells, or we can say other cell types. You are able to see a pattern of chromatin cell structure is happening. So attack data gives you more pattern as compared to the cell expression data, because that is having a very unique way of chromatin modification that is happening at cell level to generate or to get the trajectory of the cell populations. So this is going to help interface free linking of transcriptome and epigenome across thousands of cells. So instead of having lot many mathematics stuff to be done, it can be done in a very simple and easy way. And this is also one of those window of our software where you are able to see all the cell population, which we are able to categorize. And then we have just chosen one NCM1 gene, where you can see the promoter signals in the last 2 or 3 cell populations, where you can see the expression is happening because of the promoter signal. And if there is some element here which has like some bigger height in this upstream at more than, because this is this Morgan, we can say mega bases away. And you can just see the expression is very, very high in the last population, because there you can see more number of this element is present in higher percentage, we can say. And in the downstream also we have some effectors. So upstream and downstream effectors were found here, and ultimately you can see the expression was happening in these cell types. Whereas here is the inhibitory effect, where though you are getting the signal of the promoter, but these elements have an inhibitory effect. And that's why in this we have very limited amount of gene expression. And where you don't have any expression, this element of NCM1, so these cells also are not expressing the gene. So you need this to be there or captured, or the cell has to be open up for these NCM1 promoter, so that gene expression can happen. So in this way, you can have more analysis of the data, or you can find out whatever gene you are working on, how its regulatory elements are arranged or captured when we are doing the attack plus gene expression workflow in a more comprehensive way, I can say. Now, as I told, that single cell technology is also important for tissue sections, and people want to map the data to the tissue location. So why it is very, very important, if you see these two sections are ultimately the stained slides of two different tumor, and the red one and the blue one is the tumor, and the red one are the tumor infiltrating lymphocytes. So if you see from the top image, these stills are moving inside or entering inside the tumor, and which is like important to manage the disease. Whereas in the bottom one, it is in the periphery. So that means our TILs are not entering in the tumor, and tumor is like resistant to that particular TILs. So this is a poor prognosis. So we have to do some changes in the treatment so that the TILs should enter in the tumor space. Now, with example, I would like to say that what type of data you are able to generate. So this is like some portion of the paper. You can refer it from a slide that they have done some reveal the spatial trajectory of the non-small cell lung carcinoma. So what exactly they have done? So they took the tumor samples from 12 different patients, and the category was small cell lung carcinoma and adenocarcinoma. And then they did the tissue sections, then do the staining, capture the RNA, generate the data, do the clustering part, and ultimately whatever fold change of various genes were there, that was analyzed, and also do the pseudotime analysis so that they can do the trajectory of the various cancers spots that are moving or moving in that particular tissue section. In this, if you see in this figure will be like how many types of cells we captured. So first they categorize like data is coming from what sample type. So only two colors you are able to see. You are able to see the orange one is the adenocarcinoma. So almost they are clustering in this top one. Whereas the small cell lung carcinoma having a one more different cluster. So that means there are some differences. And at the same time, if you see the number of cell types that we are able to identify was almost 11, and you can see different clusters that is there. The most important part is the patient wise. When you do the patient wise cell population analysis, you are able to see like this one, the fourth one. So it has its own cell population, which is very much different from the rest of the sample types. So this means these cells or this patient have uniqueness in terms of the gene expression of the cell population that is happening. So that's why it is very, very important to understand or generate the data at the single cell resolution or 1 to 10 cell resolution, so that what is the real heterogeneity we can understand. So this is very, very important when we are trying to give the heterogeneity figure of a particular or a picture of a particular sample. Now, this is like the actual staining that was done for 1, 2, and 4. If you see 1, 2, 3, I'm not showing the 4 one is there. And where you can see in the 4 one, the cancer is moving to different locations, and the cancer and stroma is intermixed. Whereas in the 2 and 1, that sample, it is at only one location. Now, they try to dig out more information from this sample. So they did the cancer and the stroma study, and they have just cataloged gene expression pattern or specific genes that are responsible for invasion, migration, or we can say cancer stages and everything. And they just catalog the heat maps. So if you see in the cancer one only, the heat map of those genes are totally different, and because tumor microenvironment also regulates, so that's why you can see the very different heterogeneity in this all the 4 samples, all the 3 samples. In the stroma one also, you can see the sample number 2 is behaving differently from the cancer number 4 or the cancer number 1. And similarly, you can see in that particular all other genes also, you will have a different expression pattern. So that means our tumors are really very heterogeneous at individual level. Now, in this sample, what they did, some Surratt clustering analysis of the cancer areas. They also given the MET score that will give you a like invasion efficiency of those cells. What is the efficiency? And tumor plurality means how many types of cells are there in that particular tumor. So that also they were able to figure it out from the same data. Now from this, you are able to see from this cancer one, the pseudo root analysis they did, and they found out that one root there was no branching happening or not movement. So this is confined to that location only. From location two, it has moved to various location, and ultimately this one is leading to different other locations. So ultimately, by understanding this way, we are able to find out what exactly is happening at that cellular level, and gene expression level, why it is invasion migration is so much happening, and whether we can have a specific drug to stop that migration. So that was like a small portion of the publication, but I will encourage everyone to go through this publication. Now, how exactly you have to do means what is the real biochemistry behind it. So since we are capturing data from the tissue section, so we need a slide. And if you see, this particular slide has 4 capture areas in which you will be placing the tissue section. And ultimately, you can have 2 options. Either you can strain with H&E or you can strain with immunofluorescence, and ultimately you capture the image of the tissue. Then you will follow steps to capture the RNA from these tissue section on the slide itself. And ultimately, you will be constructing the library that is going to be sequenced on Illumina machine. And then once you analyze the data, then you will be using the Loop browser to visualize the data. Now, uh, our slide are very specific, where you can see there is a 6.5 by 6.5 capture area where the spots are there. So almost 5,000 spots are there, and all these spots are barcoded, and the size of every spot is 55 micron, and it has millions of primers, and each primer have a specific special barcode, the EMI and the poly-DT to capture the RNA, and the partial read one is part of Illumina library. So we are capturing the RNA from the tissue above the spot. So ultimately, this barcode data is going to map the data to the location on the image of the tissue. Now we have enabled it on a human and mouse FFP section, where by using probe chemistry, we are synthetic poly A probes are there, which will be which will be specific to uh, all the 18,000 genes in human and 20,000 genes in mouse. And using this synthetic probe chemistry, we are able to capture the data from the FFP samples. So that will be helping you to generate data for biomarker discovery, for the clinical research, and the normal and diseased study can be done from the tissue sections. Now, if you see the workflow differences, you want to see the fresh frozen tissue is directly happening. The staining and the imaging is happening here. Then polymerization is happening to release the RNA. And then cDNA synthesis is happening on the slide itself. Now the data is getting barcoded, or the molecules getting barcoded. And once you prepare the library, you will map it to the location, the data on the tissue image. In FFP, there is an intermediate step of adding the probe hybridization and ligation step. Then you will have RNase treatment and polymerization. One, the probes will be polymerizing, and these synthetic probes are getting captured and extended. And then ultimately we sequence those extended probes and map the data to the location of the tissue. This is the whole workflow of FFP and fresh frozen. So fresh frozen is a typically 1-day workflow if you are starting your OCT embedded tissue sections. And FFP is a 2-day workflow where we have overnight hybridization step that has to be done. And ultimately next day you can just prepare the samples, and you can construct the library and sequence it on Illumina machine. So this is like what exactly you will be able to do when you are using the FFP. So you will have the complete view of the disease complexity. You will be able to do new biomarker discoveries. You will be able to understand the novel cell types and their states. And at the same time, you can also see the spatial organizations and use it for creating the cell atlases. And spatiotemporal gene expression patterns can be easily you can do on a software part, where with what gene is expressed where, you can just figure it out just by searching the name of the gene. And these are the slides. You can see actual slides that are being used to do that. So your bulk data is looking like this, all jumbled up. So that has generated a lot of information, I can say, right till date. But now people use a new technology where they segregate this data into a single cell resolution, more neater, we can say, and then figure it out what is happening wrong in these various cell population. Then still, for the tissue people or tissue lovers, you want to map the data to the location. So this is the way our technology is going to help to map it to the location. And now people are combining these two data at the same time to get more functional aspects of the biology of a particular tissue section. So that because right now it's not single cell resolution. Once that we launch, then you can simply generate directly from here. And this is being supported by both the workflow, that is the Chromium workflow and the Visium workflow from 10S Genomics. So these are like where exactly we are in the clinical space. So you can do the high throughput target screening, you can do multi-omic characterization, you can do cell type and the cell state identification, the track the clonal behavior, the CAR T cell transgenic TCR, that is right now people are promising and doing this therapy, can be easily done, and then unbiased detection of antigen expression. Now, this is just a summary, I can say. So we are in the target discovery, we are in the characterization of infusion, we can do the response monitoring, and we can do resistance profiling using these technologies. And at the same time, this is the whole summary, I can see. We are in the Chromium single cell technology and Visium technology. These two technologies are in the discovery, in the translational phase. People are using it. Now, new technology is coming in situ sequencing. So in situ sequencing is going to be integrated microscopy based data generation, where multiple data from the same tissue sample can be generated. So the first two technologies is based on NGS based data generation, and this will be independent technology that is going to generate the data directly from the tissue section using in situ based sequencing method. So I'm happy to take any questions, and thank you very much. Hope I was able to make you people understand about the technology and how people are using it. So we are open for questions.
Thank you, Nitya sir, for the detailed information. So here we have couple of questions. So
Yeah.
the first question is, do we need to have prior information regarding cell size before putting it in the machine?
Cell size in general, yes, people have to, like, it should not be more than 30-40 micron. And typically those cells are in that size. And if you have a challenge, then you will have to use the nuclei of the cells. If the cell is very big, then you have to work with the nuclei.
I hope we have answered, Sagar, your question. So the next one is, can pleural fluid be prominent source for sample collection for this kind of analysis?
Which fluid?
Pleural fluid.
Pleural fluid means any fluid which has cells can be used, but from there you have to get the cell and the debris removed, and that can be used as there has to be cell in that fluid, and you have a method to purify the cells or enrich the cells.
Okay. So the next question is, in certain diseases where it is difficult to extract cells from affected area, how should one go about it? Also, what is the minimum number of cells required for effective data analysis in such cases?
When your number of cell sample is very limited, so suppose you have 100 cells, 200 cells, or like our LT kit is supporting 1000 cells. So 1000 cells to load, you need at least 3000 cell total sample. So that limited can be done. And if it is very in that way, is like at least minimum, I will say 5,000 cells has to be there so that you can start off your experiment.
Okay,
Because with very limited cells, you can do directly bulk data using some low input RNA kit. Because if you're not very much sure that you will be able to do a live cell suspension, then either try for Visium technology or try to generate directly data from some low input kit where less number of samples can be handled.
Okay, thank you, Nitya sir. Let me check if there are any other questions. Other than tissue samples, can stored cells stored at -80 degrees Celsius can be used for single cell analysis?
So because when we store cells in -80 in a RNA later, the RNA is getting permeabilized. Though RNA remain intact, but our main purpose is to map the RNA to the individual cells. So such cells are not recommended in a leaky way. We need cell population which have intact cell membranes, and there should not be any leakage of the RNA from due to storage, which is expected when we store at -80. So that is not recommended. 10x is coming up with a kit where you can fix the cells for up to 7 days in 4 degree, and you can start your experiment once you collect those cells.
So there is one question who is interested in minimal residual disease in hematology. The question is, from bone marrow point of view, it's high heterogeneity. What is the accuracy that is followed?
Accuracy that is followed in terms of if you say the rare cell population, we can pick it up. We are capturing all the RNA data of individual cells, so you are getting the complete story. It is not based on very specific marker. We miss out something. So the story is complete. I will say we are giving a complete picture of the sample. From there, we can figure it out what cell type populations are there in that heterogeneous sample. So I can say that instead of using 10 marker, 20 marker, or 100 markers, I'm giving the complete story of the samples, what exactly various cells are and what genes they are expressing. So it will be a complete story instead of a very small story using 10 marker or 20 marker.
Followed by that, there is one question more. What is the minimum sample needed?
Minimum sample, as I told, for single cell workflow, you need at least 5,000 cells has to be there because you need some QC and other stuff. That will be the starting point, I can say, because even if you want to profile 1,000 cells, at least 3,000 cells has to be there with you.
So the next question is there any screening criteria if we get huge number of cell types? How to categorize them?
How to categorize them? If you having a lot many samples you are profiling, the software is going to help to use the like clustering is happening automated way. So it is not like user determined, but there will be some software that is going to help you. But once you search like marker gene analysis, because first is clustering is happening randomly. So that is, we are not controlling, it is software driven. And then you search the specific cluster, what gene they are expressing. So because the table will be there in the software, in the Loop browser, you can go, so that will let you know what are the top gene expression happening in that particular population. Then you're able to figure out like what cell type it is based on those gene expression pattern. So number of cell is not a limit or any issue will be there. Ultimately, there should be a heterogeneity. If it is the same sample, they will be clustered together. And if they are different sample, they will be clustering into different clusters.
Okay. So the next question is what all parameters are checked in single cell QC?
What all parameters are checked in single cell QC? Number one, the cell debris has not to be there. The viability has to be above 80-85%. The total count has to be there. You should be aware that the temperature should not do the clumping of the cells, and the right type of media that you want to process it. There should not be any RNase activity of a particular cell population. So that's why you need some specific modifications, because if the cells are like neutrophils which have RNase activity, that will degrade the RNA during the sample preparation only. So those things we have to consider altogether.
Nitya, the next question is, people want to know the quantity. How much ml minimum quantity is needed to start the process, I guess?
Oh, that is like if you have like a 1000 or 500 cells per microliter, then at least 50 microliter cell, I will say 50 microliter samples will be sufficient. Even 100 microliter, 50, 100 microliter. So it is the number of cell is more important, because you can just spin down and enrich, make the stock concentrated.